I want to thank Jim and the other organizers for inviting me. I actually did not know that at the time that it was a keynote address. And I imagine that some of you are familiar with the story. I'm a physician, but at the time of my daughter's birth 10 years ago, almost to the day, it was clear that there were some issues. And it really, at that particular time in the OR, I was more a father than I was a physician, although I couldn't help but notice that she had feet, very long feet that reminded me of the Marfan, although I'd never seen an infant with the Marfan syndrome. But it progressed from a curiosity in the first week or so to some concern. And then by the time that she was about 3 months old, she was diagnosed with failure to thrive. And so we were in a mild panic, my wife and I. And the first picture is a picture of her actually taken when she was 18 months old. And she had probably seen 4 or 5 different physicians at that time, each focused on a different organ system. And it was each diagnosis that came along with that exam was not quite right. Some were good guesses. But it was clear we did not have a diagnosis. And you probably can see a few of the features that caught some people's eyes at various times. And probably her bent fingers were actually, and her toes and both hands warranted the diagnosis of arthrogryposis, which just means bent fingers in general, or contractures generally in the small joints in many cases. And one thing I didn't notice, because I have 3 children, but I didn't really notice that her eyes were slightly far apart, which developmental geneticists probably would have picked up, but we didn't see one of those. There weren't any actually in our area of California. So we were kind of stuck with no diagnosis, and we're trying to get her to gain weight, which was very challenging, and she never really did. She kept falling off the curve. And I became more skeptical about seeing more physicians. And I had this nagging feeling that really her physical exam was not complete. That is to say, nobody had really looked at her head to toe like the old-fashioned clinical geneticists do. And so I decided to take her back where I trained, and that was at Hopkins. And this is one of my favorite pictures of all times. This is Bea when she's a little bit older. And this is Victor McCusick, who trained me in internal medicine and clinical genetics. And this picture was taken about a year before he died in his apartment with Bea sitting on his lap. And I had taken her back because I wanted, as I said, one of those old-fashioned diagnoses or old-fashioned physical exams that might lead to a diagnosis, because that's really what we were in search of. And in this picture, you probably can see she has blue sclerae, which is very unusual for somebody her age and very indicative of connective tissue disease, although the classic osteogenesis imperfecta she did not have. She had a number of other facial findings, which if you're a geneticist will kind of stand out. And I had called Dave Valley, who I didn't know very well, who was a pediatrician at Hopkins. I trained on the adult side, and the pediatricians didn't talk to the adult people back when I trained. And he was very gracious and arranged to have a fellow examine Bea, which he did, and he called in somebody else, and they looked down her throat, and they were kind of all shaking their head. And what they were looking at was her uvula. And her uvula, which is that little thing that dangles in the back for no apparent reason, was split. It had a little cleft in it. And this was a really important clue as to what Bea might have. And they called in another visiting faculty member named Bart Loeys, and Bart examined Bea and concluded, based on the hypertelorism, the eyes wide apart, and the bifid uvula, that she must have what we now call Loeys-Dietz syndrome, which is a mutation either in the TGF-beta1 or TGF-beta2 receptors. the TGF-beta receptor 1 or TGF-beta receptor 2, which are heterotetramers that transduce signaling from either TGF-beta 1, 2, or 3, the ligands for those receptors. And by chance, I had already scheduled B to have an echo at UCSF, which is close to where we live, a couple days later. And the reason that was important for the diagnosis of Loeys-Dietz is the third feature of that disease is aortic dilatation. And fortunately, she did not have any abnormalities of her aorta at that time or at any other time. And before I had that echo, Bart Loeys gave me a paper that was three weeks old describing the syndrome that we now know that bears his name. And the average age of death was 26 years. And so B and I are flying back on this plane while I'm reading this paper, and I'm slightly crushed by this because this whole process had been one of, did she have a mitochondriopathy, which most of those of which are devastating diseases, or did she have something more benign? And that physical exam, though, the discovery of the bifid uvula, in addition to some of the other features that B had, suggested that she had some kind of abnormality in TGF-beta signaling. And in fact, I put together this diagram because this is the Marfan syndrome, and some of the things that B had are in blue here. And she had some features of all of these diseases which are allied in that they are TGF-beta signaling in some way or another is being affected by fibrillin 1 mutations, fibrillin 2 mutations, the TGF-beta receptors themselves, and in this case, the TGF-beta 1 ligand. So it seemed as though B probably had something going on in TGF-beta. This was really her set of findings, but the most prominent thing for her was her muscle weakness. She was, from a motor point of view, delayed in every single way, and she still remains weak compared to her peers, although she's adapted quite well. And she is slightly hypotonic, which has its own problems when you're trying to ride a bicycle. But these other problems are, at this time, are more of an inconvenience. The flat feet and the joint laxity and those kind of things are not things that really require attention. What's left out of here with Loeys-Dietz and Marfan is aortic disease, and obviously, that's even though she has a normal echo now, it's always a possibility. And so I went from really being sort of a father to then a clinician trying to figure out what she had diagnostically to drawing the conclusion that in fact she had probably a new syndrome that involved TGF-beta signaling because clinically she didn't fit into any of those other categories, even if you allow for allelic variation and quite a lot of variation within mutations in one particular gene. So this is the dummies version of TGF-beta signaling, and this comes out of the Science Pathways site, and it really probably represents about 10% of the proteins that are actually involved in TGF-beta signaling, which is why I call it the dummies version. But this was really where I started thinking biochemically about what's going wrong with B. And just to back up, TGF-beta, and of course there's 1, 2, and 3, which is not recognized here. TGF-beta is bound to, by a series of other proteins, to fibrillin, which is part of the extracellular matrix. That's the problem in Beals and Marfan. And then Loeys-Dietz is a mutation either in ALK5 or in the TGF-beta receptor 2 itself. This is otherwise known as TGF-beta receptor 1. This was the other problem with coming into this field, is that everything had 4 or 5 names. And it took some doing to try to decipher all this, but this is a problem that we all have. So I began to think, okay, if she doesn't have mutations in ALK5 and TGF-beta 2, which were the first that were looked at, maybe she has some problem in all these other things. So I actually read a review by Se-Jin Lee, who was a friend from Hopkins, who had discovered myostatin, which is one of the TGF-beta ligands, one of the 33 members of that family. And because it was called myostatin, it kind of caught my eye, and it regulates muscle size. And I thought, well, maybe by analogy with the TGF-beta receptors, which are gain-of-function mutations, this is the receptor, as it turns out, these two are the receptor for myostatin. These are conveniently called activin receptor 2 and 2B. The A is actually not really used. So this associates with ALK4, and it also associates with ALK5 on occasion. And so I decided that probably B had a gain of function in the myostatin pathway in the signaling domain of the kinase domain of these receptors. And I tried to get people to sequence it for me, and because of IRB rules and that kind of thing, nobody would do it, so I sequenced it myself. These three genes, and not surprisingly, it took me quite a while, but what took me more time was actually analyzing the sequence, because I was deathly afraid that I was going to miss a single nucleotide. Almost all these diseases are single nucleotide chains. They're all dominants. Many of them are new mutations. To make a long story short, they were completely normal, which I thought was unfortunate, because I thought it was such a great hypothesis. And I'm sure there are kids out there who might have hypomyoplasia owing to defects in these genes, but not Beatrice. So some of this work I did borrowing pipettes and that kind of thing from Andy Fire, who worked at Stanford, was a friend from Baltimore. And Andy at that time was working with various genome companies, evaluating their equipment, and he said, well, why don't you just sequence 100 genes? And I thought, well, that's a clever idea, because I have at least 100 here that I would like to look at. But that turned out to be difficult to do, to look at 100, but it's a lot easier to look at 10,000. And I just happened to be at a meeting where I bumped into somebody I had known from long ago, and that was the CEO of Illumina. And that was when Jay Flatley ran molecular dynamics, and I had worked a little bit with molecular dynamics back in the late 80s late 90s and around the 1999, 2000. I hadn't seen Jay in 10 years, but I asked him if there's anybody at Illumina who would be interested in sequencing B, because I was pretty convinced at that time that she had a genetic disease. And it probably had to be a new mutation, or it was a recessive. But because all the other diseases in this family are dominants, I figured it probably was a new mutation, probably for me. And he introduced me to Gary Schroth, who was at that time working on transcriptomes. And he said, sure, I'll do it for you. So he did the whole family. And there's nothing really involved in TGF-beta that we could discern. Of course, we only had 55% of the exome in that sample. But actually, we picked one that we thought was the most likely. This was a gene called CPNE1 that seemed to, at least in somebody's hands in Switzerland, had some influence on canonical TGF-beta signaling. And we presented a poster here in this very meeting probably 6 or 7 years ago showing that. And it seemed plausible at the time, but I wasn't 100% convinced. And so I actually looked at about 400 chromosomes looking for the allele frequency of this, because it wasn't in the database at the time. And sure enough, there was the homozygotes were about 1% of the population, and I figured this just can't be Beatrice, because she's unique. And we would have seen this before probably. And so I was a little bit down in my cups again. And I went back to Gary and I said, can you do anything else for me? And he said, well, we're working on this chip that would capture exomes, and maybe we could do that for your family. So in order to test out the hardware, we ponied up some more DNA, 5 of us. And I told Arena, who was the person who did all of the computational work, and I worked with her for about 2 years looking at that transcriptome. It was really a lot of work because the software was not very friendly. I was constantly pestering her. So she was used to me. But I said, if anything with TGF-beta pops out, let me know. And a day later, she sends me an email saying, well, there are only 2 genes that are mutated in B that are unique to B and not in the rest of the family. And one of them is in this gene called TGF-beta 3. So I said, that's probably got to be it. So the process was, from that point on, not only verifying it, but then the question became, well, so what? You've got this mutation. What's it doing? And perhaps this doesn't surprise this audience, but the literature was 20 years old for the most part on TGF-betas. As you can see, a lot of them didn't even bother to differentiate between the 3 isoforms, beta 1, 2, and 3. And so I basically made pilgrimages to all the people who were in the Cold Spring Harbor monograph on TGF-beta, including the editor, Rick DeRink, who happened to be at UCSF. And he was kind enough to give me actually his the draft copy of the monograph, which I obviously devoured. And then I set out to talk to as many people as I could who had anything to do with TGF-beta signaling, particularly in muscle development. And honestly, there wasn't very much, but I got lucky. A person I'd done a postdoc with in Seattle named Andrew Lassar, who had discovered MyoD, I went to him and I said, what do you think? And he said, well, I don't know anything about TGF-beta, but I know this other guy that does. So that turned out to be Malcolm Whitman, who was a couple blocks away from where Andrew was. And I went over to him and I told him the story, and I said, do you think you could help me understand what this gene is doing? And so he said, sure. So we made a bunch of constructs, and another friend of mine put all those constructs into human cells and assayed the activity of TGF-beta 3 wild type in the mutant version. And Malcolm put them into xenopus eggs. And it's a great system for studying TGF-beta, and we concluded that it was acting as a dominant negative. The TGF-beta ligands are dimers, homodimers, and the mutant ligand was completely dead. And when it was co-expressed with the wild type, there was diminished activity versus wild type alone. So we concluded that B had a dominant negative, but also it was likely that the phenotype was owing to decreased TGF-beta signaling. It might seem obvious, but there was some, and still is, some orthodoxy that any perturbation of TGF-beta signaling, as in receptors that I just mentioned, mutations in those receptors, actually result in a compensatory overexpression. But our data was fairly clear that we were dealing with a hypoplastic, hypomorphic mutation. And just to show you, this is the sort of the critical part of the TGF-beta. This is the so-called Cysteine Knot, which defines all 33 of the ligands in this family. They all have these 4 cysteines here, and then in the donut hole there is another disulfide bridge with 2 cysteines, and then the 7th cysteine holds this monomer to the other one, so you have a dimer ligand. And B's mutation was in the cysteine to a tyrosine. And there was abundant literature to show that that all of these actually were critical cysteines going back to worm. And so it was time to completely switch gears. We published a paper, a case report this summer on B, and primarily with the hope of actually trying to beat out of the bushes other Bs. And one possibility is that she has a relatively benign course, and little girls and boys who are as skinny as B grow up to be Twiggies, and that's it. Or that they have vascular disease later in life and it kind of is not connected to their phenotype or whatever. So it does require that we have yearly echoes just to make sure, because I think that's going to be her primary major problem. So our family is a post-genomic family. We live in that world of what's going on developmentally. In terms of developmental biology, it's very clear that this problem was, this hypomyoplasia was active in utero because of her fingers and that kind of thing, and you can date the timing of the creases. But I think that the genomic field is really still at the observational sort of stage where we're just observing genotypes, genomes in general, their structure, et cetera. But at some point, all of these hypotheses that are generated by genomic information have to be studied in model systems. And that's clearly what we're doing with B. This is a picture of B with a mouse that has her allele knocked in. So she's probably the only girl that has her own mouse. This mouse actually lives at home. And I can tell you, they produce an amazing smell. So I force her to keep her door shut. But she's pretty wedded to that mouse, I can tell you. And so that really winds up the story because we need to find more bees and we need to study this mouse intensely. This mouse is very new. This was taken just a couple weeks ago. So we're just in the process of back-crossing it and getting the right background and then really studying it. And I think that's where all of this genomic work will eventually have to go, is really testing hypotheses and put things into model organisms. So I'm out of time, so I'm going to say thank you very much for your patience.
Thank you very much, Hugh. Questions from the audience?
Go ahead.
Thanks. So Hugh, you mentioned that early on that an IRB policy hindered an early attempt to sequence a candidate. Could you expound on that and explain a bit more about that part?
Yeah. To friends of mine who had the capability of sequencing couldn't because it was clinical material and the information was going to be reported back to me. And so, to a man, they said no. So that's... Now, I got it done, probably extracting DNA and doing various things in various labs around that I borrowed probably violates some rules as well. But there's nobody here who knows about that. So.
We won't tell. Other questions? All right. Oh, we one here.
Yes, thank you. And because when you talk about the TGF-beta signaling path, actually I work some data for Parkinson's disease. So actually, when you talk about this pathway, just remind me, once I actually find some, TGF-beta signaling pathway has some interaction with the Parkinson's disease. So because Parkinson's disease is also kind of, within the muscle, I think has some issue with the muscle. I'm not quite sure. I just kind of, because this TGF-beta is so significant in my study for the Parkinson's disease. I'm not quite sure if there's any relationship.
Is that? Are you saying Parkinson's disease?
Yeah.
To be honest with you, well, cognitively she's completely normal, so that's good. And her motor function, her fine motor function is good, and she doesn't have a tremor. And she did have an MRI when she was young, although she was very young when she had that, so there's nothing there. But the TGF-betas are doing all kinds of things. And I don't think we have, we know 10% of what we need to know about TGF-betas. And that's not just during development. There's a postnatal component to TGF-betas. And involved in the immune system, et cetera, et cetera. So I wouldn't be surprised if it was, I don't know about Parkinson's because I would have thought that would have come up in some of these larger screens that have been done with those patients. But it wouldn't surprise me that it's involved in some trophic fashion.
Actually, I have
Yeah,
I'm sorry, is there a question somewhere else?
You mentioned that there was 2 mutations.
Yeah,
What was the
she
other?
also had a mutation in. Yeah, the other one's de novo as well, which is about the right number, for an exome. She had another mutation in a gene. It was. I forgot. But it's been well studied in mouse, and hers was a stop codon, codon 63. So it's clearly a truncated, probably just a loss of function, complete loss of function, null. That heterozygous null mouse has completely normal phenotype. So I'm assuming that it doesn't. It could be a modifier. It's always possible that it's a modifier. And probably with this mouse, I'll backcross it into that mouse just to see. But yeah.
I actually had a 2-part question, which is, you obviously sit on the perch of, as you said, of both a physician and a parent, and with access to wonderful brains and tools through your professional network. But, we all want to also think about the other 99.999% of people, and I think that's one of the reasons I would imagine that motivates you to tell your story, is the other. So I guess I have a double question.
It's a good question.
Yeah, so my first part is, we're beginning to see a lot of these episodic impressions from an awful lot of people.
Mm-hmm.
And the question becomes, how can we aggregate this community to have a unified voice? Because it seems to me that a lot of policy is driven from people who might be serving those people, but not, you know what I'm trying to say.
Yeah, so let me answer the first part of the question first. And I get this question a lot. How generalizable really is this story? And I would say that what is common with me and a lot of other parent advocates, let's say, or patient advocates, as the case may be, is it's more about persistence and trying to eliminate the asymmetry between what I know and what the experts know. And I might be able to narrow that a little faster and with somewhat greater facility. But you'd be surprised actually how little, I mean, there is still a huge, huge gap. And if you combine everything that everybody knows about TGF-beta and then what's not known, we're all in the same camp, really. Because everybody has these model systems. I'm speaking specifically about this, but I think it probably applies to a lot of situations where the vast majority of the literature is using these very, very context-dependent bioassays for TGF-beta activity, et cetera, and they don't really translate to the human. And if I don't get a phenotype in this mouse, I can live with that, because a mouse is not a little girl. But I think what all these parents have, and I would be included in that, is at least some of them anyway, they're just persistent. They will not take no for an answer, and if the doc says come back next year, that's not good enough. And so I think trying to reduce that asymmetry, which many parents do by just reading, and sometimes they sound foolish. I've seen lots of those kind of parents myself, but I never dismiss it because it's an effort on their part to try to become a peer, if you will. And that's all I really tried to do. I never really pretended that I was a genomics expert or a TGF-beta expert or anything else, but at least I could have a conversation and get somebody interested in it, and that's really what parents do.
So just to follow up on this, though, and so I can sort of set a little bit of maybe spirited discussion, given that we're opening the meeting, is some of our colleagues, if you were not driving this, but you were a passive participant of this, would not have returned this finding to you because they might have perceived that it did not meet burden of proof to be significant and clinically actionable. Comment.
Yeah, well, it's very clear that this is not actionable. What I didn't say is that based on a mouse experiment that somebody published giving anti-TGF antibodies and also using losartan to tamp down SMAD2, SMAD3 signaling that had dramatic effect on those mice. And I treated B when I was not quite certain about that, and partly because there was also a paper, or several papers, showing that angiotensin receptor blockers like losartan had an effect on skeletal muscle. The drug is very safe, and I decided unilaterally, really, to treat her and see if it would have an impact on her muscle. It didn't at all, and she was on that for 3 years. And when I first, so there was some actionable component to that, but you could question whether that was sound or not. You know, I don't think it put her in any danger. But most of the time, it's not actionable. But what I would say to that is, as a parent, it doesn't have to be actionable. For me, the first step is really a diagnosis. I want to know what it is that we're dealing with, because I can't get a prognosis or a management plan if I don't have a diagnosis. Now, you can deal with little symptoms and things like that, but that's not very satisfying.
Yeah, I guess my question is going one even further step back, which is a lot of people may or may not consider, depending what data they have access to or what their biases and preconceptions are, they might not even consider this a diagnosis. In fact, somebody might say, oh, this is just another variant of unknown significance.
In a way, it's not a diagnosis. It does
Sure.
put you in that family of diseases, and then you can say to yourself, well, if I was doing surveillance on her, you know, I should make sure she didn't have scoliosis, and I'd check for all the other things that those other children have. So it's a quasi-diagnosis of sorts. I mean, the fact is that what you don't want to have happen is to get T-boned by some, you know, catastrophic vascular event when they're 18, and you could have looked for it when she was 12. That's what you're really trying to avoid. And when I first set out on this, my goal was to look for things that were actionable, that you could do something about to avoid that kind of catastrophic outcome. And it turns out she didn't have anything horrible like that. But I think that's got to be the first workup, and you eliminate those things, and then you go after some of the other things.
Two quick things. One, I definitely would have tried the losartan. Two, has anybody reported mutation in this gene since your paper came out? Three, have you looked through any clinical databases?
Past short period of time, but I've called a lot of people.
Okay.
And said, I sent them the paper and said, could you look for these kind of people, and asked some of the gene diagnostic.
Going the opposite way, look for mutations in big clinical programs, and then see what the patient looks like clinically.
The only one where that's been done, and there are new mutations, no reported mutations in the coding portion of that gene, was a big cohort studied by the NIH on connective tissue disease. But nothing yet. So, but there's got to be kids out there with that mutation. There's got to be. My hope is that they're all, they all get to be 80, and that's it. Nobody hears from them because they're just skinny little people.
Thank you very much, Hugh.